Hello students, here I am to continue the life history of Pyrosoma. In the previous lecture I had told you about its structure, mainly the structure of the colony, and in the colony reside thousands of individual animals which are very small in size, and they in turn with their reproductive activity give rise to new Pyrosoma, and the colony as a result goes on increasing. So let's begin with the further with the animal Pyrosoma. Now here is the diagram of the single individual, which actually is the animal Pyrosoma, and these are arranged in the colony. Like you can see, this is a type of section of the colony. This is the outer area, the ocean in which the colony is living, and this is the test around the animal. The test, it is making a test process which is covering the branchial aperture of the animal, and this is the inside of the colony with the as I had mentioned, the common cloaca, and this is the atrial aperture. So all the waste water along with waste material, it comes and it is poured in this common cloaca, and this common cloaca, as you have done before, it opens to outside by a common cloacal aperture which is guarded by the velum on the periphery. So now let's do the detailed structure of the single individual. Now the alignment of the animals, it is the animal's antero-posterior axis. This is the anterior end and this is the posterior end. The branchial aperture is present on the anterior end, here it is the branchial aperture, and the atrial aperture is present at the posterior end. So this axis, which is passing through the anterior, through the body to the posterior point, is the antero-posterior axis. The antero-posterior axis of the animal is at right angles to the surface of the colony. This is the surface of the colony, and this is the antero-posterior axis. Okay. Now the dorsal part of the animal, this is the dorsal part and this is the ventral part. The dorsal part of the animal, how do you recognize the dorsal part? The dorsal lamina is present on the dorsal surface of the animal, and endostyle is present on the ventral surface of the animal. So this dorsal part, it is facing the open end of the colony, like the common cloaca. If we visualize the whole colony, like all the individuals, they will be, one will be here, this is the one will be here, and so on. So the whole colony, its closed end will be this side. This is the common cloaca. Closed end is this side, and the open end will be this side. So all the dorsal surfaces of the animal, they are towards the open side, and the ventral surfaces, they are towards the closed side. And so this was type of alignment of the individual with respect to the colony. Now let's do the internal structure. Now we start from here, the branchial aperture, which is guarded by a test process, means it is covered. Branchial aperture is not wide open to the ocean, it is covered by a test process, and this test process is made by the test of the adjacent test lying adjacent to the animal. And this branchial aperture, it is on the inside on the inner side is having the tentacles, which are known as the branchial tentacles, and these tentacles, they filter whatever it is entering into the branchial aperture and coming inside the body. After the branchial aperture, we can see there's a cavity, a narrow cavity, which is having muscular bands. This cavity is kind of the cavity present before the pharynx. Like you can say it is a prebranchial cavity, and the branchial cavity is covering most part of the animal. Most of the space inside the animal, it is covered by the branchial cavity, and connecting both the prebranchial and the branchial cavities is in between these two cavities is the lining of peripharyngeal band, and this peripharyngeal band is having two luminous organs situated on it, which are responsible for luminescence, for emitting light. This animal, as you know, is known as Pyrosoma. That means the fire animals. And this the illuminated organs, the organs which emit luminescence, are responsible for the name of this animal. On the peripharyngeal band, on its dorsal side, is present the dorsal tubercle. Here it is the dorsal tubercle, and a nerve ganglion, even dorsal to the dorsal tubercle, and an otocyst. These are kind of sensory structures. On the ventral side of the branchial cavity is present the endostyle, and the branchial cavity whole is having big stigmata like this. The horizontal openings, they are the stigmata, and they are crossed by the longitudinal bars which are present inside the pharynx. So these are the longitudinal bars, and these are the stigmata. Stigmata, they are labeled, this is the one, this is the one, and like this. So all of these are long stigmatas. The branchial cavity, and one more thing, on the dorsal side, the dorsal lamina, as you can see, along the dorsal lamina there are some mesodermal mass present. These are mesodermal cells which are also responsible for formation of these luminous organs, and the dorsal lamina is lining the dorsal surface of the pharynx, and dorsal lamina has many long processes hanging inside the pharynx, and these are known as the languids. And these languids, they are responsible for catching the food particles, whatever particles will come inside the branchial aperture along with the water. The water, it washes the pharynx very well, and it selects the food particles and sends them upwards towards the dorsal lamina, and by the help of these languids, these particles trapped in a cord, in a mucus cord, they are passed down, and when they are passed down, they enter the esophagus, because the pharynx cavity, it is continuing into the esophagus. So the food, it travels along the languids and it enters the esophagus. Now the whole alimentary canal, it is consisting of a pharynx and then esophagus, stomach, intestine and anus. It is present on the rest, apart from the pharynx, the rest of the alimentary tract is present on the posterior side of the body, where the atrial aperture is, because the anus has to open to outside, but the colony, it does not open directly to the outside. So the anus, it opens into the atrial aperture, and the atrial aperture, it opens into the common cloaca, and all the waste material which is coming out of the animal is washed by the water in the common cloaca to the outside. After the alimentary canal come the gonads, and the gonads are a continuation of the endostyle on the posterior side. Endostyle is present ventrally in the animal, so the ventral endostyle, on its posterior most edge, the gonads are present, the testes and the ovum. And endostyle is also responsible for growing stolons, which in turn develop buds, and these buds, they develop into new animals which keep on adding into the colony. So in this way, this animal, it gives rise to new individuals by budding. Further, this is all the description that I have explained to you. The closed there's a bilobe mass of round mesodermal gland cells. This is the source of luminescence. The rest of the alimentary canal, this is the alimentary canal. Okay, so we've done all this. Now about the one more thing I forgot. Along with the alimentary canal and the reproductive organs, there is also a structure, heart, which lies immediately behind the endostyle. And there's a multi-lobe testes and a small ovary with a single womb. Position I told you, and this you will have to memorize, that the testes is multi-lobed, and ovary, it at a time produces only a single ovum, and they are lying posterior to the alimentary canal on the ventral side. This I've shown you in the diagram. The gonads develop from a cord of germinal tissue which forms a part of the stolon or epicardium. The latter is a ventral continuation of the pharynx and endostyle. Pyrosoma is protoendous. Protoendous means the male organs, the male reproductive system, it matures first, and the female reproductive system follows afterwards. Fertilization is endogenous, it is happening inside the body. Segmentation is meroblastic. You must be knowing what is meroblastic. The egg is having a large amount of yolk. Due to having a large amount of yolk, it does not undergo any division, and the only disc, the blastodisc present above that yolk, the divisions are only limited to that disc. The development is direct without a larval stage. The segmentation results in an elongated embryo which develops into an oozoid or cythozoid. Here you can see the structure of a cythozoid. From the stolon, a new individual which is formed is known as the cythozoid. The latter gives out a ventral stolon which later divides into 4 pieces. Means the cythozoid, it gives out a stolon on the ventral side, and this stolon, it develops into 4 acidozoids or blastozoids, and till they are fully developed, the cythozoid keeps its existence, and after that it finishes itself. After the cythozoid is finished, the tetrazoids, which are having the ilioblast cells, these ilioblast cells are responsible for the formation of all the organs in these acidozoids or the blastozoids. And these 4 acidozooids which are attached to each other, 4 in number, they form the tetrazoid colony. The young tetrazoid leaves the parent colony, its cythozoid gradually degenerates, but the 4 tetrazoids give out ventral stolons. Now, cythozoid had given out one stolon which forms the tetrazoid. When the tetrazoid gets degenerated gradually, then they become free. The tetrazoid colony becomes free, and now each acidiozoid or blastozoid, it gives out a ventral stolon, and by budding they keep on adding individuals to the colony. So this way a colony is formed. So this was the life history of Pyrosoma, and to summarize, the acidiozoids, they give rise to a cythozoid, and then the cythozoid, it gives rise to a tetrazoid colony, and acidiozoids, they are sexual phases, and the cythozoids are asexual phases. The acidiozoids, they give rise to the cythozoid by the interaction of ova and testes, and the cythozoid, it vegetatively produces the tetrazoid colony. So this way there is alternation in sexual and asexual phases. Thank you, and in the next lecture I will start with Salpa.